Beep-a-dee-ba-dee. Beep-a-dee-ba-dee. All right, everything seems to be working. All right, good morning, everybody. I'm just kind of hanging out here with one of my pups. There she is. Sentinel pup. Good morning, everybody. Yeah, happy Friday. I got to say, I am very, very, very glad I am not riding my bike into work this morning. I would quite literally freeze my buns off. So sitting on my couch at home is far more comfortable. All right, while we wait for people to roll in, let me get the chat up. Anybody got questions so far about anything? Oh, looks like Michael was in space again. Hey, Jana. Or not?
Good morning, Professor.
Good morning. All right. So today we've got to finish up retinoic acid signaling. There's kind of some main principles that need to be covered, I guess, which will help you understand what's going on. It's not, hello, Tanya. It's not a trivial thing to get, particularly because it's very different to everything that we've come across so far, right? And so in lots of different ways. And also, it's kind of confusing because it's both something that works at a kind of a global level in the sense of the organism, and also it acts at a local level, right? So if you kind of roll back the clock to many moons ago when we talked about types of cell signaling, I put retinoic acid under endocrine. Well, kinda, right? So I was kind of right and kind of wrong. In some senses, it does behave as an endocrine signal. In other senses, it behaves as a paracrine signal, right? So that kind of gets at this complexity, right? And how different it is from everything else. So I'm just going to get my computer set up for, oh, yeah, it's telling me it's dead by right now. Using the whiteboard. Put it on my little dinner snack table thing. Get that out of the way. If any of you have, I'll move this over a little bit. I've got space. There he is again. Any of you have issues kind of reading what I wrote, or if it's too dim, reflections, so on and so forth, give me a shout, let me know, okay? Because this is going to be non-trivial to do on a small whiteboard. All right. So I'm actually kneeling on a cushion here. Isn't that funny? See, hello. Hello, Sophia. All right. Retinoic acid signal. My dog's looking at me wondering what I'm doing. Oh, Nomi. So really there's two major parts to RA signaling. One is the production of RA. Right. Second is, oh, hang on, production and degradation. This is where it gets so complex. So I was just checking to see who's here. Oh, I hope Elaine shows up. Oh yeah, you are there, Elaine. Sorry.
I'm here.
Yeah. I was scrolling too fast. You're right underneath my symbol. Okay, so production and degradation of the signaling molecule itself, right? That's one part. And the second part is the mechanism of RA. Ah, shoot, how do you call it? I don't want to say signaling. Yeah, that'll do. Kind of doesn't really quite say what I want to say, but it'll do the job. That's my phone. All right, so to get how this all works, you have to really get those two parts and how they interact, right? Because you can't get the whole picture without both of those pieces, right? It just won't make sense. And in many ways, this is the most important. Crap, got to take another picture now, haven't I? This is what establishes the signaling gradients and tissue specificity, and also when this happens. So in many ways, how RA actually does what it does is of less interest than how its production or how its levels are controlled. Right, because the level of RA is essentially the sum of, or the balance between production and breakdown. Right, it's kind of a
Dr. Crook. I'm sorry, can you repeat it's for tissue specificity and what else?
Temporal. So when it gets expressed and where. And actually that's get a fancy word up because we all love fancy words. So spatiotemporal is really the word that's bandied around a bunch when we talk about RA signaling. So essentially you have two variables where it is expressed or where it acts, really is a better word, and when it acts. Because RA signaling acts multiple times during development and in multiple different places and tissues and organs, right? And so that's what makes it such an astonishingly complex pathway to study. A very difficult one too. Right, take another picture. I'll delete the other one. Okay, so RA production and degradation. Shit, I can't stretch that far. There you go. Ta-da. It's a good job I've got long arms. So the beginning metabolite is vitamin A, also known as retinol, if you're being fancy. And this is produced by the liver. and it's secreted into the blood. So I'm not entirely sure if vitamin A and retinol are exact equivalents, or vitamin A is processed a little bit into retinol by the liver. I think they're the same. I've found them being used interchangeably. So I'm not entirely sure what the liver is doing. No one ever talks about that. They just go, it's produced by the liver. Anyway, so retinol, vitamin A, and so obviously this also depends in part on your diet, right? Because this vitamin A is something that you get in your diet as part of your diet, but also I think it's synthesized from other stuff. I just don't know what. That's biochemistry. Anyway, so this is floating around your blood. Doobie-doobie-doo, mind its own business. And it gets taken into a cell. Let's just put it here. Right, so this isn't the cell that's receiving the signal. This is a cell that's actually involved in the production of RA. Right, so the two things are spatially and maybe even temporally separate. This is what kind of fudges that distinction between endocrine and paracrine signaling. And in that cell, and is taken in by a protein called retinol binding protein, which you don't need to worry about too much. It is going to be converted by, and I'll write these out in full in a second, actually. Do that because it's a reversible reaction. By one of two enzymes. These are called alcohol or retinol dehydrogenases. There's an S at the end of that, promise. See? And there's a bunch of these. Funnily enough, ADHs are also what your liver produces when you go on a bit of a bender downtown. So when you consume ethanol, alcohol dehydrogenase is what do they convert it to? Something else that is less toxic. Anyway. That's a reversible conversion into retinaldehyde. Now, ADHs and RDHs, these are present in most cells. Oh, yay, the trash is getting picked up today. Right, so the tissue specificity does not come really from these. Right, everybody following along okay? Don't want to outrun the class.
So far, so good for me.
Super cool. Oh, got someone in chat. Give me a second. Good. Now, the last step to produce retinoic acid and I do apologize for my handwriting. It's not good at the best times, but when you're kneeling next to your couch, it's even worse. This is an irreversible conversion catalyzed by RALDH, which stands for retinaldehyde dehydrogenase. Now you can see why we shorten it to RALDH. Get a different color. Okay, run out of space. And the round DHs, there are several of them. Can't actually remember how many. Actually, I can check, can't I? I've got the paper open that I was reading. Where does it say? Man, there's so much information in here. It's hard to actually find the information that you want. There's 3 ADHs, 2 RDHs. Oh, there you go. 3 retinal dehydrogenases. Thought it was 3. Let me just close some stuff so I can get you lot back. There we go. So I was just checking my facts. Now those 3 different round DHs are expressed in different places and at different times. So it's really this part here which gives you the specific, well, this part of the production leads to specific patterns of retinoic acid production. Now I've got to check myself because I almost told you something that wasn't quite right. You know, don't want to do that. I'm just going to get rid of this because I need the space. And I'm going to extend that cell membrane out there. Now, retinoic acid, it's not just about production, it's also about degradation. So I don't know what it actually gets broken down into, little bits and pieces. Retinoic acid is also the substrate for enzymes called CYP26. And there's a bunch of those too. These are, I get kind of frustrated at how little space I have. So if you hear me grumbling and grind my teeth and stuff, that's why. These are cytochrome P450. Move that over there. So these are enzymes. There's actually a whole load of them, because we've got like at least 26 different ones, which are involved with breaking stuff down. They're actually produced a lot by the liver, but they're also produced by other cells. Their main job is to basically degrade stuff, not just signals, but all kinds of metabolites. And so these cytochromes reduce the amount of retinoic acid produced by converting it into a inactive metabolite. Couldn't tell you what that is off the top of my head. Okay. That's a lot to take in. Yep. Drink. Yeah. Questions?
So the RALDH, is that an enzyme?
Yes.
So that enzyme that's the one that's like responsible for specifying all the types of tissue or cells?
Right. So the reason why I caught myself is because the amount of RA present is the sum of how much RA is produced and how much of it is degraded. right? So that's why I have to be very careful with language, because there is a lot of spatial specificity and temporal as well, spatial temporal even, right? Of RAL-DH expression. So what tissues and when these enzymes, and which ones of them as well, right? Because there's 3 different RAL-DHs. Oh, hang on, I'll get to the chat question in a second. Be there in a moment. But also where the degrading enzymes and when these are expressed also affects the levels of retinoic acid and the gradient that results from that. Right, and so that's where this gets really complex. Does that make sense, Michael?
It makes sense. So what I took away is that there's a lot of factors involved
Right,
in
yeah.
specifying more
Yeah,
than just the RAL-DH.
Correct. Yeah, so it's both, it's a balance between the production of the signal and also its degradation. Now let me get to Peyton's question. Oh, right. So Peyton, CYP26, the substrate for CYP26 is retinoic acid. And so CYP26 converts retinoic acid, the active signal, into something else. What that something else does, I think it actually does nothing. I think it just gets, you know, recycled, consumed, kicked out of the cell, whatever. I don't actually know. But it no longer plays a part in retinoic acid signaling. And this is irreversible. It's one way. So once retinoic acid has been degraded, it doesn't go back into this loop. It's just done. Tanya, yes, it's all chemistry. That's why I get a little bit like, ooh, when it comes to chemistry, because that's not my strong suit. So cytochrome P450, that's a class of enzymes which basically convert something into something else, right? Typically a degradation step, right? They take a metabolite and they break it down. There's another case, sometimes they turn into something useful. I'm trying to plumb my memory of, I know there's one of these in the DAF signaling, TGF-beta signaling. I can't remember if it's involved in the production or the breakdown of one of the intermediates. Anyway, it's not super important, but essentially this is in RA signaling. This is something that kind of reduces the amount of signal. So everybody got this okay? I'm going to wipe it off. Okay. So let's say you have, I don't know if this is going to help. I'm just making this up as I go. Yeah, that's right, Peyton. Correct. So let's say we have a lot of RA production. Right, and remember, RA is a diffusible signal. It's actually diffusible not just between cells, but actually through cells as well. Now, if you just have, you know, a lot of, pardon me, RA production, right, over time, right, you'll get just loads of it all over the place. And Elaine, this actually reminds me of a question you asked a while back, I think based off of one of the quiz questions. It's actually stuck in my head. So you do get some degree of gradient here, right? Because obviously this cell, if this is the, oh crap. Hopefully that washes out. There we go. Right, if this cell is the RA producing cell, you'll get more retinoic acid around here than you will over here, just by simple physics, right? Takes time for it to diffuse. And also it's not going to diffuse forever, one presumes. I don't know. Anyway. Getting myself distracted. Now, if you have other cells here, and I'm just labeling that's not the enzyme in the extracellular space, right? So these enzymes act within the cell that they're expressed in. They're not secreted. I'm just to make that clear. Now what happens is that retinoic acid gets taken up by those cells and destroyed. Right, like so. And we're still getting retinoic acid produced over here, but it's also being degraded over here. And so now what you end up having is this gradient. Oh, that's not very good. Established of the signal itself. So in other examples that we've looked at, the signal, or at least of a paracrine signal, the gradient is established purely by where that signal is produced and by diffusion. In retinoic acid signaling, the gradient's established as a consequence both of production of the signal and its degradation. And I think in many ways, this is how you can establish very robust, stable gradients in development. So it doesn't matter how long this cell is going to be producing retinoic acid for, you're not going to change the shape of that gradient, right, as those retinoic acid molecules diffuse, because they're always being degraded by other cells. Get my hand up there. So that's what makes it very suitable for setting up gradients and maintaining those over developmental time.
So are you saying that like this is why RA signaling is a good model, or are there other gradients established like in a similar way?
As far as I know, and that doesn't necessarily mean a whole lot because, you know, I'm not all knowing. Retinoic acid signaling is the only one where the gradient is established both by the production and degradation of the signal. There may be others, but scraping through my brain, I can't think of any. I mean, in some sense, I'm thinking about RTK signaling, but I think it's also the case for others. ligands are removed by the act of reception. So when they bind to the receptor is usually endocytosed, which removes the signal. But that's not really a mechanism for establishing the gradient. In the case of RTK signaling, the gradient is simply the diffusion. And also, here's another kind of, like aspect to it as well. RA is small and very diffusible. All of the other signaling molecules that are diffusible that we've talked about, and even some that we haven't, are proteins. They are much less diffusible. They're physically larger. It takes them longer to get somewhere. So if you think about it, and I'm not making this up on the hoof, but I'm just, thinking about it as well. That would explain why retinoic acid signaling relies on both degradation and production to establish a gradient. Because it's so diffusible that it could probably fail to establish a gradient simply because it moves fast enough away from where it's produced that a gradient won't establish. Does that make sense?
Yes, it does.
Yeah, it's like you'd swamp the tissues, kind of, in a way. Yeah, so that's just another way of looking at it. Okay, all good? I'm gonna get rid of this. Okay, so that's all about, and it's really worth spending a lot of time on RA signaling because it is such an important molecule in a lot of different developmental contexts. We'll see it kind of over and over again throughout the course. And that's also, if you think back to those different phenotypes of excess retinoic acid signaling that we looked at on Wednesday. That's why it also matters a great deal when the embryo is exposed to excess retinol. Because most of those embryos miscarry. But it depends on when during development the fetus is exposed to excess retinol. If it's very early on, most likely because of the role of RA early on in development, development will be so badly affected that it will result in a miscarriage. If it's a lot later on when, body plan and tissues have been already sorted, then it may affect kind of development of individual tissues like the eyes or the ears, things like so. Anyway, so, and there's actually I'll have to dig out the picture from the paper that I was looking at, which was really neat. So essentially you have, let's use colors. Now we're talking about the nucleus, and you have genes which have upstream of them in their promoters things which are called rares, which is RA response elements. Rares. And so, hey, Tanya, you remember genetics? This is how eukaryotes coordinate gene expression. Actually, Michael too, you're also in genetics, right? They do so by having are DNA sequences in the promoter regions which are recognized by different transcription factors. So when those transcription factors are present or activated, depending on, which one we're talking about, they're able to bind all of the genes, right? And there are lots that respond to RA. All of the genes which have these rares. And retinoic acid signaling can both activate and also suppress gene expression. I'll get into why that's important in a little bit. Anyway, so oops, sorry, these tables are a little wobbly. In the nucleus, we have, these are typically dimers. It's not super clear exactly how these work. Don't fully understand it, to be honest. This is a little bit of a blunt pen. Like so. We have two proteins. These are together the receptors. That's what RAR stands for. Oh, yeah, sorry. Actually, in my defense, inism is like really far away from my hand, and so it'd be hard to write. So even if I did write it, it probably wouldn't make any sense. Sorry, didn't catch that. So normal, or not normal, but other signaling pathways, the receptors is kind of over there, sticking out on the surface of the cell. Retinoic acid signaling, because RA is lipophilic and scoots across membranes willy-nilly, the receptors are actually proteins inside the nucleus. Which is pretty weird to think about, right? So we have retinol being taken up by the cell, and there's all kinds of funky business that is tied in with that. And maybe it's degraded by CYP26. But if it's not, if this is a target cell, right, it goes into the nucleus, binds to RAR. Might even bind to this one, but really, I don't want to get into the weeds here because it's so freaking complicated and confusing that I don't think I'll do a very good job. I'll just confuse you. And that then is able to bind to these rares. Like so. Now, one of the genes which is upregulated is, unsurprisingly, RALDH. And can't remember the number, I think it's 8. I'm just going to do this right now. One of the genes that's downregulated is a gene called FGF, right? I think it's FGF8. What's it say in the PowerPoint? It doesn't, bugger it. So, not really much more to it than that, really. Essentially, in target cells, let's put this. It goes into the nucleus. In non-target cells, it's degraded by CYP26. So it doesn't go into the nucleus, doesn't have any effect on gene expression. Gotcha.
So it's kind of like with the notch signaling. I think it's notch. Like it goes in to the nucleus and then it wants to produce more notch, so it can keep like producing the signal, right?
Not 100% sure how analogous those is because I don't fully understand how this ties in. I just know that that's one of the. I would hesitate to say it's the same sort of positive feedback mechanism because it's very clear in notch signaling that that is what happens. Here, it's just so much more complex. I don't want to, be definitive about it.
Okay.
Because there's also kind of sequential events. So you get, you receive RA signaling, you turn on a particular gene pattern. Part of that may be promoting RA signaling through a different RALDH at that particular locus, which then establishes a new gradient over developmental time, right? And so it's just too complicated to really do a good job for me to do a good job of explaining it. I'm sure that's not much of a consolation.
Yeah, thank you.
Yeah, this is actually I think it's by far the hardest signaling pathway to learn about. But it's also, in many ways, one of the most consequential. Everybody done with this? All right, so
Dr. Crook.
hello. I hope you're not going to ask me a question about what I just rubbed off.
Maybe. So, the RA signal, when it's outside the nucleus, can get degraded by the CYP26, but also as it gets to the rare and increases the RA LDH, that RA LDH can also get degraded by the CYP26.
So the CYP26, as a general rule of thumb, will be expressed in non-target cells. So I'd actually have to look. It's quite possible. I'd have to check, but conceptually, at least, it makes sense if CYP26 was one of the targets for downregulation by retinoic acid signal. So any cell that receives the RA signal that's a target cell further suppresses the production of the thing that breaks down retinoic acid. Does that make sense? Yeah, don't worry about it. But yeah, I kind of took a shortcut because I had a limited amount of space, but CYP26 is something that's expressed in cells which are not, I guess one could call it competent to receive the RA signal. So in other contexts of signaling, competency is about the expression of the receptor, right? In retinoic acid signaling, that's not the case because the receptor, the actual proteins in the nucleus, are always present. In
Okay, so
RA,
basically,
Go on.
the CYP26 has more affinity for non-competent RA signals?
no, it's more that it's produced in cells which are not, it's kind of what defines competency in RA signaling. So a cell that doesn't express CYP, actually, let's write this down. Make it clearer. What color did I use? I think it was this one. So this cell would be not competent. Right? By definition of it expressing the enzyme that breaks down RA. So even though it can take up RA, that RA will not make it to the nucleus because it will be broken down. Whereas cells which do not express CYP26 are deemed competent. Because any RA that gets into them, which it will just by diffusion and also some active transport too, will make its way to the nucleus and bind to those retinoic acid receptors.
So cell with CYP26, without it, it's competent.
Correct.
Okay.
With it, it is not.
Mhm.
So you see, kind of weird and wacky, right? It's complete opposite way around from other signaling pathways. Now, one thing that I want to quickly cover, because we're going to lean on it later on in talking about different stuff, is RA doesn't always act kind of alone. It interacts with other signals. And so if I can remember, do I have a green? Oh man, there must be like some kind of shortage of green pens. See if I can remember. This is in the mesoderm, right? So this is kind of later on in. Those are not, that's not a 6-pack, just in case you're wondering, because this is a very early stage embryo. These are called somites. You don't need to worry right now, but you will come across them later on. And we have, this is anterior, this is posterior. Kind of, pretty shitty drawing to be honest, but it'll do. And so at the anterior, and this actually in many ways, retinoic acid signaling is really big in anterior posterior patterning. It just doesn't necessarily have to do it over the whole embryo. It actually can do it very locally, which is kind of weird. So I have a lot of RA signaling here. And remember that FGF that I mentioned being one of the targets of retinoic acid signaling, but to downregulate it. FGF is, I think it's FGF8, but I'd have to check, might be 6. FGF is a posterior patterning signal. So FGF signal is, oh, I'd have to double check. remember what type of signaling pathway that is. I think it's a TGF-beta type signal, but don't worry about that. So these two are antagonistic. What that means is that wherever you have FGF signaling, you have no RA signaling. Wherever you have retinoic acid signaling, you have no FGF signaling. It's kind of funny to use the word antagonistic in a biological context, right? They antagonize each other like siblings. And so the consequence of that is you have a retinoic acid gradient like so, and an FGF gradient like so. And so not only does retinoic acid signaling establish its own gradient based on production and degradation, but also through the effects it has on gene expression, it affects the production of other signaling gradients. So now instead of just having one piece of information, now you have two. How much RA are you experiencing and how much FGF are you receiving? And so the kind of classic genetic way of drawing that would be like so. RA signaling suppresses FGF expression, and FGF suppresses RA signaling, if I remember correctly, through the upregulation of CYP26. Ta-da! Plenty of food for thought. All right, just because we're actually at time, but if anyone has any questions, feel free to shout them out. I'll leave the Zoom running so that anybody who has to go can catch them. Oh, patches, my knees. Oh man. Ah, here I am. Stretch some cricks out of my back and my neck. Oh. All good? Make perfect sense?
Dr. Crick, are we still having the quiz on Monday? No change on that?
Yes, and in class 2. If anyone still has issues arising from being in quarantine, stuff like that, that can't make it to class on Monday, just let me know and we can arrange another time for you to take the quiz. I did think actually quite hard about doing it online, but it would completely mess up with the style of the quiz and what I'm hoping to achieve with it. Because it being a kind of a make sure everybody's in the same place kind of deal, if it's open book, then you can just look this stuff up. So yes, Peyton, everything up to and including what we just covered. And again, it's not designed as a punitive, like, haha, gotcha kind of deal. The whole point is to make sure that you get all of this, because I can't emphasize how important it is to get all of this, because we're going to be kind of starting away at the races after this. This is our kind of gentle introduction. It's very important that you're all, kind of on top of what we've been talking about. So that's the aim of it. Any other questions? Yeah, I'm taking that wasn't a question, Elaine, but a commentary.
Yeah. You asked if everything was clear, so I said clear as mud.
Clear
It's
as
a thing
mud.
my dad says.
Awesome. Yeah, I like that one too.
I'm a little foggy on rares. It's been a while since I took genetics.
Mhm.
If you could, do you have any
So-
time to clarify?
Yeah. So response elements is a catch-all term for a regulatory DNA sequence, right? And so let's say my usual example is heat shock response, right? Because it's pretty well defined and it's pretty neat. So let's say, a cell is undergoing heat stress for whatever reason. There's a whole bunch of different genes that need to be turned on and expressed for that cell to survive that, right? That could be proteins involved in protein misfolding, others involved in, cholesterol production, right, for cell membrane fluidity, all kinds of stuff, right? So to turn all of those on in a coordinated manner, rather than go like having a transcription factor specific for each one, what you have is the same sequence of DNA in the regulatory promoter of all of those genes. So you only have to have one protein that recognizes that sequence. And whenever that protein is present, all of those genes get turned on. That's called a response element. And so you have those for really anything that has any kind of coordinated response, which, to be honest, is an awful lot of genes processes. Right, so if you want to turn on a developmental program for, I don't know, lung development, let's say, there's a whole ton of genes which are going to be needed for that in different aspects, different ways, right? And so all of those are going to respond to like the lung specific transcription factor or collection of transcription factors. Now, essentially, if you don't have that transcription factor present, those genes don't get activated. So that's one way of getting tissue and cell specificity and gene expression, right? So all of the genes involved in lung development are on in the lungs. They're very much off in other tissues. So, Mackenzie, the same DNA sequence. So essentially rare retinoic acid receptors, those RARs or RIXs, those recognize a specific DNA sequence. And that DNA sequence is going to be upstream of whatever genes need to be expressed in response to retinoic acid signaling. So the DNA sequence and the protein kind of match. I got to do my twisty business because I'm too lazy to get off the couch now. So essentially, let's say we have a rare here and a heat shock response element here. Sorry, it's really hard writing on a whiteboard at 90 degrees. Oh, hey, no worries, Tanya. You too, check up on this if you need any further clarification. It'll go up on YouTube in a little bit. So any gene that has that rare, and there'll be multiple genes, right? When retinoic acid is present, bound to these, that will bind to them. Does not bind to any other genes. Right. Now, Ba-do-ba-do. Wish I had more colors. Actually, what am I talking about? Fucking, sitting on them. Sorry, dog, don't worry.
Could you say the thing that you just said a minute ago? Any gene that has that rare when RA is present. What was the rest of it?
Will respond, will be bound by this. So when retinoic acid is present, this protein and retinoic acid will basically seek out any bits of DNA that it recognizes, these rares, and it will bind to them. It will not bind any other genes.
Okay.
So if the role of this is to turn on or off a gene, it will only turn on or off genes that have rares. It will not bind to genes which do not have rares, right? That may have other response elements. So we've got heat shock protein, or is it? No, that's something else. Right, heat shock protein, when it's induced by heat, because it's changes shape with temperature, will bind to heat shock response elements, but it will not bind to rares. It's really, in kind of simple terms, a way of coordinating gene expression of a lot of different genes simply by the presence or absence of the transcription factors. So if this is present, only genes which respond to this will be expressed. No other transcription factor present, no other genes expressed other than the ones that have rares. Does that help?
Does help.
Yeah, it's kind of weird to think about because really this is just a DNA sequence. But transcription factors are DNA binding proteins, that by definition. So these DNA binding proteins recognize a very specific sequence. They will not bind anywhere else. They don't bind randomly. They're very, very, very specific about the sequence they bind to. And they typically only bind when they have the correct shape, right? Which is what retinoic acid binding is all about. It will change the shape of this so it now recognizes the DNA. That's all right. You're welcome, Mackenzie. It was, I think I taught that, didn't I? You were in my genetics class? I can't remember. Yes, but it will only bind that sequence. It won't bind any other sequence. So if you want to get a gene expressed in response to this, typically all you have to do, oh, I'm sorry, Mackenzie. All you have to do is put that DNA sequence in front of that gene and it will get expressed. So if you took this DNA sequence and you put it here, in addition to this one, this gene would then be expressed in response to both of these because it would have two rares. It would have two response elements, sorry, a rare and a heat shock response element. Yeah,
You
sorry.
said that they're specific, right? For the
Very
protein
specific.
that they're binding.
The protein only binds specific sequences so that the two go together. The protein that binds them and the sequence that the protein recognizes.
So the heat shock protein wouldn't bind the rare when it's next to it.
Nope.
Okay.
But that also means, and this is why this is worth kind of a good point that you brought up, recapping, you can change where and when in response to what genes get expressed by moving these things around. And one of the examples,
sorry, go ahead.
so one of the examples I love talking about because it's really well studied, which I'll talk about later in Evo Devo, is the evolution of wing spots in butterflies. Essentially, wing spot production in butterflies is because an eye pigment gene is now expressed in the wings. And they think that the reason for that is that gene, eye pigment gene is in every cell, right? Every cell has the same DNA. So they think that the way in which that happened was that a wing specific response element ended up inserted in front of the eye pigment gene. And that was selected for because it provided protection against predation. So yeah, there's a lot of expansion of function based on movement of these response elements. It's pretty funky. Yeah, Mackenzie, I'm really sorry about that. I've heard that same thing from other students, unfortunately. The good news is that he's no longer teaching anything to do with genetics, particularly after last semester, which was a giant shit show. I might have to delete that from the recording, sorry. So yeah, not much consolation, but if there's anything that kind of you need going over that will help you with this stuff, let me know, like as and when it pops up, and I'll go through that so you're not struggling. Cool.
I just want to clarify really quick. So the rare, the response element is like a this may not be the right word, but just so that I can get through my sentence, it's a kind of like a precursor to activate a certain subset of genes that are next to it.
Yeah, pretty much. Yeah, precursor isn't quite the right word. Tag, maybe,
Oh, okay.
or switch, right, is another way of thinking about it. It's a switch that only RXR can turn on, or RAR. And so if you don't have the finger flicking the switch in that cell, that switch doesn't get switched, doesn't get turned on.
That's cool though, that it can turn on like a whole set of genes as opposed to each one of them having to get turned on individually. Very
Exactly.
efficient.
And many genes have multiple response elements. So it's not like a single switch, it's like, 10 different switches, which control when and where and in response to what that gene gets expressed. It gets
right.
pretty complex.
Yeah.
Mhm. Biology is always complex, right? Kind of like the default state. Cool. Anybody
Wow.
else have any questions? Yeah, we'll hit upon this stuff many times over this semester. That's for sure. Cool. All right, buddy. I'm going to go make myself a cup of coffee. If you have questions before Monday, email me and I will answer them as quickly as I can. I have a fairly chill weekend, hopefully. Pardon me, this weekend. So if there's stuff as you're going over it in preparation for the quiz that you need clarifying, just let me know and I'll do that.
Thank you.
Cool beans. You're very welcome. Enjoy your weekend and I'll see you on Monday.
See you Monday, Professor. Bye-bye.